feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong, in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong, in my happy place. And I'm right where I belong, in my happy place. And I'm right where I belong, in my happy place.
Hello, everyone. Welcome to the National University of Singapore Yonglou Lin School of Medicine Healthy Longevity webinar. Thank you so much for joining us today. As you can tell, I am not Professor Brian Kennedy. I am Belinda. But don't you worry, today we'll have another professor. Actually, we'll have two professors joining us later. One of them will be moderating the questions later with us. But today we have a very special episode because we have our own research assistant professor, Jorming Goh, to join us today. So I'd like to remind you to use the Q&A function to send any questions. We love receiving and hearing from you. And before that, we are going to have Ian Chu, a research associate at NUS. He will present his research team study on comparing DNA age estimates from EPIC V1 versus EPIC V2 chips in blood and saliva.
Thank you for your introduction. Good evening. As mentioned, my name is Ian Chu, and today I'll be presenting our recent comparative analysis on DNA methylation age estimates across the Illumina EPIC V1 and EPIC V2 arrays by using the barcode, peripheral blood mononuclear cells, and saliva samples. DNA methylation is a cornerstone of epigenetic regulations and has emerged as a robust proxy for estimating biological age, which what's often referred as DNA age. Over time, aging leads to predictable site-specific changes in DNA methylation patterns at CpG dinucleotides across the genome. DNA and biomarkers leverage these patterns to quantify aging at the molecular level. Traditionally, these biomarkers were built on platforms like HN450 and EPIC V1 chip. In contrast, the new EPIC V2 array offers expanded genome coverage and enhanced detection of enhancer, but retains only about 83% of EPIC V1 probes. This updated design may introduce bias in estimation. Another consideration in DNA amine measurement is the difference between the blood and saliva types, considering the cell compositions are different across the samples. So, our main objective was to evaluate the agreement between the DNA age derived from EPIC V2 and EPIC V1. We compared these estimates across three different sample types, which are barcode, peripheral mononuclear cells, or PBMCs, and saliva. Next, I'll briefly describe the design and methods we employed. We performed a cross-sectional analysis within the APA trial. 16 healthy participants, aged 40 to 60 years, provided the fasting blood including the barcode and PBMC and saliva samples. DNA was extracted from the samples and profiled using both EPIC V1 and EPIC V2 array. We estimate DNA age using 11 DNA biomarkers, which includes 6 PC-based and 5 non-PC-based biomarkers, all under consistent normalization and imputation methods. Next, onto the demographic of the populations. Our 16 participants had a median age of 48 years, were predominantly female and Chinese, which represented a relatively homogeneous cohort. This table shows the number of CpG sites in DNA and biomarkers, and sites not retained in EPIC-V1 and EPIC-V2. In EPIC-V2, CpG loss ranged from 3 to 10% for biomarkers like Howard and phenol H, rose to 15 to 25% for Greenwich and DNA and T telomeres, feed age, and ink biomarkers. This loss were concentrated in gene bodies, intergenic regions, and CpG islands. Next, this heat map shows the Spearman correlations between the DNA MH estimates from EPIC-V1 and EPIC-V2 across all 11 biomarkers and 3 different sample types. Most of the PC-based biomarkers exhibit very high correlations above 0.8, indicating robust consistency. However, some non-PC biomarkers, particularly in saliva or certain PBMC measures, fall below 0.7, suggesting that these biomarkers are more sensitive to the prop losses observed in EPIC-V2. Next, the box plots display the absolute difference in DNA MH between EPIC-V2 and EPIC-V1. In contrast, PC-based biomarkers such as PC Hannum, PC Greenwich are much more stable, with median difference often below 1 year. Saliva stood out here, with both of the widest variants and largest median sheaves, likely due to sample heterogeneity. This consistent sheaf, always in the same direction per biomarkers, suggests systematic rather than stochastic bias driven by the array design and prop loss. In conclusion, although DNA MH show high correlations between EPIC-V1 and EPIC-V2, significant and systematic difference in estimated DNA MH do occur, particularly for non-PC biomarkers and in saliva samples. Our findings highlight that direct comparison of DNA MH across platforms should be made with caution. For longitudinal tracking or intervention study, especially for those small effect size, the bias could meaningfully distort the interpretations. So moving forward, better cross-array harmonizations and tissue-specific deconvolution strategies, especially for saliva, will be crucial for accurate DNA and profiling. I would like to thank to my supervisor, Prof Andrew Meyer and Dr. Weilan Wang for their guidance throughout this project, Mr. Dejian Hua for co-writing this research paper, and special thanks to the entire team at HLTRP program. Thank you for your attention.
Thank you so much, Ian, for that insightful study. It's very important for us researchers to really think about those potential biases. All right, so without further ado, I'm going to introduce our guest speaker today. Dr. Goh Jorming is a research assistant professor and the principal investigator of the Exercise Physiology and Biomarkers Laboratory at NUS. For the past 20 years, he has done extensive research in exercise physiology in both preclinical animal models and human studies. And today we have quite an exciting talk from him about the role of exercise, specifically Exerkines. So over to you, Jorming.
Thanks very much for the kind introduction, Belinda, and it's wonderful to be back here on the show. What I will do next is I will go ahead and share my slides. And thank you very much again. I would like to talk to you all about how exercise is medicine, and it's a very important topic, especially for healthy longevity. All right, and then there's going to be I'm going to allude to the role of exerkines in my talk. All right, so just quoting this quote from, or getting this quote from Nish, that which does not kill me makes me stronger. And it's really illustrating the concept of hormesis, where when we talk about certain organisms or certain substances or practices that actually causes some kind of harm to the organism, it actually allows the organism to get stronger. And really, exercise is part of that hormetic mechanism that actually allows us to get stronger with more doses of exercise. So before we even begin, I think it's quite crucial to actually define what exercise and physical activity is, because they tend to be thought of as a single entity, but they're quite different. If we were to just strictly define them, physical activity really is any body movement that's produced by contracting skeletal muscle, and it increases energy expenditure above basal levels. Now, when we think about physical activity, it can be broken down to different subsets. We have recreational, which is where we think about exercise. There's transportational, there's occupational. And then when we think about all of these factors, we realize that exercise is a defined subset in that it actually has a goal orientation. It actually is meant to improve fitness and different aspects of health. So exercise is structured, it is repetitive, and it's simply not just equated to be physical activity. So if the definition already completed, I think we can actually look at what the WHO tells us. And it's simply that we need to accumulate about 150 to 300 minutes of moderate intensity physical activity a week. And the problem is, if we look across the Singapore population, and even though the data here is slightly dated, the effects, or rather the data itself, hasn't really changed that much. If we just zoom in on the middle-aged adults and the seniors, you can see that for both men and women, about half of us actually report that we are not physically active when we look at regular or frequent participation in physical activity. Now, if we were to just take a time machine and go back in time, and then we can actually look at hunter-gatherers, which we descended from, but we probably can't do that. So we have to take lessons from modern-day hunter-gatherers. And I will share with you two lessons learned from the Hadza tribe from Tanzania and the Semani tribe from Bolivia. So these are pretty active hunter-gatherer tribes. And essentially what they do is they actually accrue 2 to 3 times more daily physical activity compared with most modernized societies. If you look at just the top column, if you look at the top column that shows physical activities in minutes and moderate to vigorous PA, moderate to vigorous physical activity in minutes per day, you can see that they already accrue about 115 to 120 minutes a day. All right, and that is literally the weekly load that is recommended by the WHO. So when we compare that with all the other countries on this list, you can see that there's Japan being compared, and there's the United States. They perform several-fold higher in terms of physical activity a day. Now, if we were to look at some of the health outcomes, we can see that at least in terms of compared to the other populations, the coronary artery calcium scores, which are known as CAC scores, are essentially extremely low, okay, especially at at advanced age, 70 years and above. So it does go to show that with them adopting a very physically active lifestyle, it is very well correlated with vascular health in later years. So the message is that truly exercise is medicine, but then the problem is very few people in modern society take the prescription or even meet the minimum recommended doses. Then moving gears, we're actually going to talk a little bit about the pillars of aging, specifically focusing on inflammation. All right. Now, inflammation is actually one of the drivers of aging. And we know that we see all these kinds of messages out in the popular press where we hear messages such as exercise boosting the immune system. Now, the immune system does play a role in inflammation. And essentially what happens is when there's an inflammatory event that happens, what happens then is immune cells are being recruited to an area of damage. And because of the recruitment of immune cells, they typically release substances that are messengers that communicate with other immune cells to patch up the area or to recruit even further immune cells to tackle the damage. So those are really the essential hallmarks of inflammation, where it's a constant turning on of the immune system without having it being turned off. And when we look at the modern society today, there are 2 major components that are driving the epidemic in healthcare. So one of which is obesity, and the other one is physical inactivity. Now, both conditions can actually drive what is known as a chronic low-grade systemic inflammation. Okay, essentially essentially fat cells, which are also known as adipocytes, and also white blood cells known as immune cells, can release these chemical messengers that can then, in the case of adipocytes, can block the uptake of sugar or glucose into skeletal muscle, which can then exacerbate the condition where skeletal muscle is unable to bring glucose into the body, resulting in insulin resistance and ultimately driving type 2 diabetes. And on the same hand, immune cells that are being recruited into the coronary blood vessels can actually lead to dysregulated metabolism of lipids and ultimately drive hardening of blood vessels, also known as atherosclerosis. Now, in 2005, two eminent scholars and professors, Mark Febrile and Bente Peterson, had a very provocative hypothesis, and they had shown that in many, many studies that actually when we contract skeletal muscle, when we move our bodies, when we exercise, this contraction-induced release of specific chemicals such as myokines can actually demonstrate that these molecules are effectors of muscle health or, in fact, systemic health. So they then thought that, well, then in that case, skeletal muscle should be considered an endocrine organ. And an endocrine organ really is an organ in the body that can release hormones, peptides, or different molecules that can have effects on other distant organs and tissues. So because of that, more and more research is being known in the last 20 years where we now know that myokines, which are short for muscle-derived cytokines or different messenger molecules, can thus affect different parts of the body, including the cardiovascular system, including diseases such as cancer or conditions such as a loss of muscle mass in sarcopenia or even in the brain. And that is why we consider exercise to be medicine. And that because of this aspect where muscle actually talks to other organs, we now know that there's a different slew of different identities of these proteins that are involved in these kinds of adaptations to exercise. Then the story gets a little bit more complicated then when we realize that it isn't just skeletal muscle that actually releases these potential chemical messengers. Other organs such as the liver, such as the kidney or the lungs, or even the pancreas also release such molecules. So collectively in 2016, Professor Mark Tarnopolsky actually coined the term exerkine to show that it is actually the release of these chemical messengers in the body that actually affect all the different types of adaptations that actually occur and actually contribute to the benefits of exercise. Now, to illustrate this point, a study done about 3-4 years ago by the group at Stanford and led by Professor Michael Snyder showed that up to 16,000 molecules that are being released after a single session of exercise. And in the top right corner, you can see the study design, about 36 individuals who actually brought in to perform an exercise study where they exercised on a treadmill and blood was collected before and after the exercise and then probed for different aspects of the release of different exerkines. And it showed that amongst them, there were different molecules involving either nucleic acids or proteins or even metabolites and some even lipid species. So in other words, this world of exerkine is truly tremendous, and we are only starting to probe the importance of each, or maybe collectively, all of these different exerkines working in concert together to improve health. So as I was saying, a lot of these molecules have now been shown, and these exerkines have been shown to actually have important use in disease treatment. So some of them have been identified to be considered exercise mimetics, in a sense that a lot of pharmacological effects are similar to medicine, where they have impacts on, let's say, insulin-sensitive tissues in the case of diabetes, or let's say in the case of cardiovascular disease, they will have effects on ameliorating conditions such as atherosclerosis. And there is this actual interest or of immense interest where the pharma industry is actually interested in identifying different proteins or peptides, and that could eventually serve as drug discovery targets. So in a nutshell, what we do know at least is that long-term exercise reduces inflammation. And we know that it's through part of it is through the release of such molecules induced during exercise. I just want to share one paper in the literature, which was done at Ball State University. And this study essentially showed that people that actually engage in lifelong aerobic exercise, so for example, running, cycling, and swimming, have shown to have an anti-inflammatory effect in their blood. Okay, so what happened was a group of young exercisers in let me just pull up my laser pointer here. So a group of young exercisers were compared to older adults that were lifelong exercisers that either were in a performance group or in the functional lifelong exercise group, and then also compared with another group called the old but healthy individuals. But I think the important message that we take away from this data set is that one of the most potent inflammatory cytokines, interleukin-6 or IL-6, if we look at the concentrations in young exercisers, these are just healthy young individuals, is about 0.9 picogram per mil in the blood. Okay. Now, if we look at lifelong exercises and we just look at a combined pool of those that are in the performance or functional group, it's about twofold higher at 2 picograms per mil. And then finally, when we compare it with the old but healthy individuals, it's about 3.9 grams, 3.9 picograms per mil, I apologize. So about another twofold higher than the older adults that perform lifelong exercise. So taking away the message from this study is that if we compare people that perform at least a lifetime of exercise, they are shown to have a lower concentration of inflammatory cytokines, and in this case, interleukin-6, compared to those older adults that are healthy. Of course, this is a sample size where there isn't any individuals that have disease such as type 2 diabetes, cancer, or cardiovascular disease. So one would imagine that the concentration of such inflammatory cytokines would be much higher. Now, our lab actually was interested to see what other kinds or what other types of exercise could actually modulate inflammation. So we actually embarked on a study to look at whether low-intensity aerobic or resistance exercise, which is essentially strength training, can actually modulate the inflammatory proteome. So we wanted to look at a panel of about 90 inflammatory mediated proteins in the blood. And we also wondered whether it would have an effect, or whether age, chronological age, would have an effect on this. And we were particularly interested to study this in women because they are typically underrepresented in most of the scientific literature. So a pretty big team actually participated in this study. It was led by our senior fellow, Dr. Lim Zixiang. And essentially, the team managed to recruit 16 women aged either 21 to 35 years old or more than 60 years old and were postmenopausal. They were healthy but sedentary, non-obese, and also non-smokers. And the study design was such that they came in for a baseline visit where they performed aerobic exercise on a bike, rested for 30 minutes, and eventually proceeding to do a series of different leg exercises that were meant to test their strength. Subsequently, for 2 other visits, they either did a moderate intensity cycle exercise for 30 minutes or a resistance exercise using, again, the lower limbs for another 30 minutes. And these two study visits were separated by at least 1 month. Now, the important thing to note is that before and after exercise, all the participants consented to give some blood. And then we probed about, again, as I was mentioning, about 90 different inflammatory proteins in the blood before and after the exercise. This is just a table illustrating the different characteristics of our participants. I think I just wanted to stress that they were exercising at relatively different workloads, even though we prescribed the same intensity for them. So VO2 at the aerobic threshold was significantly higher in the young, and also for the anaerobic threshold was much higher in the young compared with the middle-aged. Now, I think one of the most interesting things that we found was that, first of all, there was definitely an age effect. If you just look at one of these panels here, FGF21, we saw that there was a significant difference in terms of the release between young and older adults immediately after exercise compared with one another. There was also a further significant difference in terms of the type of exercise that was performed. So a difference between the aerobic and the resistance form of exercise. And this was pretty much consistent for the rest of the other proteins that we found. We also looked at some of the ratios of pro and anti-inflammatory responses to exercise. And again, if we were to just look at interleukin-6 relative to MCP-1, there was also a exercise type dependent response. And if we were to look at interferon gamma and interleukin-4, there was a age related difference. So this tells us something about even at low intensity exercise that there was this modulation or differential modulation of inflammatory signaling proteins. And again, we wanted to look at these kinds of impacts after an acute bout of exercise, which means what does the immediate impact of exercise do to inflammation? And this was what the intention of the study was. We wanted to look at an almost immediate impact of one session of exercise. Now moving on, we had a different study where we looked at the effects of exercise intensity and also chronological age on blood levels of this myokine or exerkine known as irisin. Now in this study, we went the opposite way. We recruited young men aged in the 20s and also middle-aged men, which had an average age of 55. What they did was they had a familiarization session on one day. They came in at visit 0, one of the baseline visits, to measure what is known as the VO2 max, or we call it VO2 peak. And then subsequently for visit 1, 2, and 3, we gave them different intensities of the cycle exercise. And similar to the previous study, we actually, again, obtained blood from them at rest, so before they commence any exercise. And then immediately after exercise, we also collected another vial of whole blood. And then 30 minutes after exercise, the final blood was actually obtained. So it was intended to see what were the dynamics of the response of this protein irisin to different intensities of exercise in young compared with middle-aged men. Now, what's interesting in this study was also we added a different component. We had all of the participants being connected to a gas analyzer where we measured the oxygen uptake and carbon dioxide production. But at the same time, we also attached this near-infrared spectrometer to the exercising thigh. Now, with this technology, what we were trying to do was to look at the oxygenation level in the skeletal muscle. So what this tells us is whether the amount of oxygen consumed by the whole body and through the mouth over here, is it also reflected at the cellular level in the skeletal muscle as picked up by this device. Now, some of the results, what we show is that at least at baseline, the middle-aged man and the young man displayed similar concentrations of irisin. And when we look at the different intensities of exercise, low, moderate, or high intensity exercise, first of all, there was a very small sample size, so we didn't see significant differences in terms of the actual area under the curve for both the young and older, albeit that the middle-aged individuals did seem to show a decrease in the irisin concentration throughout the entire exercise session, be it for low, for moderate, or for high intensity exercise. And I think what was most fascinating was if we just were to look at the compare all of the three graphs, it appears that at moderate exercise intensity, this was where the concentration of irisin seemed to be the greatest difference between the young and the older subjects. Now, we then ran some correlations, okay, between the irisin concentration and some of the other physiological parameters. And we found, interestingly, that irisin was positively correlated with this variable known as VE-VCO2, which is essentially the onset of anaerobic threshold. So it was a very strong signal. It was the R here correlation R was actually, sorry, Pearson's R was actually 0.85 and significant at a 0.05 level. We also see that in the opposite in the young subjects, the young individuals, where it was actually VO2, systemic VO2 that was actually correlated, highly correlated with irisin rather than in the older adults. Some other correlations that we found, which were also very interesting, was that the muscle correlation, the muscle percentage oxygenation was highly correlated with, again, irisin concentration in whole blood overall in all the subjects. But this was mostly driven by those in the middle age group rather than in the young, because there was no effect of there was no correlation between irisin and the left muscle oxygenation in the young, but it was a highly positive correlation between the middle-aged individual irisin concentration and that of the muscle oxygenation in. So really what we take away from this is that, first of all, we realize that there is an age-related and also an exercise-related inflammatory response to any session of exercise. And this then gives us a lot to think about in terms of how we should actually be prescribing exercise for individuals. An example of this would be if individuals were meant, for example, if individuals are diabetic, then it means that maybe a low dose of low intensity exercise may be insufficient to actually release some of those beneficial anti-inflammatory molecules. And it also means that some of these molecules may need to reach certain threshold for them to have any kind of medicinal effect. Now, if we look at the long-term impact then, the question is, what kind of exercise training then would be necessary to actually be able to reach what we call an exerkine sufficiency? Meaning that if we were to look at a certain threshold of exercise, or rather a threshold of exerkine that is necessary for health, then are individuals actually exercising enough to actually produce that kind of a response? And the jury is out. I think we don't have enough studies to actually understand this. And these kinds of studies are also very, well, I would say that they are also very lengthy and would actually require a lot of resources to actually understand this. But in any case, I think the understanding now is that if exercise is truly medicinal, then I think we can have a role to actually probe, at least in the inflammatory side of things. And I kind of end off with this quote from the late Professor Stephen Blair. He's a leader in our field. He mentioned that physical inactivity and low levels of cardiorespiratory fitness are perhaps the greatest threats to health in the 21st century. And with that, I thank you for your time and for listening to this talk. Thank you very much.
Thank you, Jorming, for your beautiful talk. I'm actually replacing Brian today. I'm Vincenzo Sorrentino, an assistant professor here at the Center for Healthy Longevity. So it's not that Brian got suddenly younger, but I just had to replace him. But I know Jorming's works and is very interesting. And I wanted to open with a couple of questions, Jorming. So first of all, yeah, based on the last quote that you just mentioned, we're talking about health and exercise, right? So it's clear that the benefits of exercise on health overall and the correlation with healthy longevity, maybe a bit of a tricky question. So what is your opinion on how exercise would actually impact life expectancy in people, right? I think I've seen small studies linking it to mortality and so on, but what is your opinion about this? Is there really a chance that exercise would really increase your life expectancy as well?
I think that's a great question, Vincenzo, and I think, we've been trying to answer this for decades, right? I think some older literature suggests that there is no impact of exercise on maximum lifespan, but I think in terms of the average lifespan, that it does have an impact on increasing average lifespan in most individuals. And I think it's mostly driven towards avoiding the onset of chronic diseases, cancer, heart disease, and so on. And if we were to look at a paper, I think it was in the British Journal of Sports Medicine. I think it came out about 2-3 years ago. They essentially looked at comparing the hazard ratios, or rather the survival curves of Olympians or former Olympians, and compared to the age-matched people that lived in the general population. And they do find that at every age level, right, that there was this increased survivorship in the Olympians, even though they may have stopped training, even though they may have stopped performing the types of exercises that they used to do when they were competitive or when they were professional. So some level of exercise may be retained throughout lifespan to actually affect some long-term benefits. And I think, again, we still don't know what those mechanisms are.
Right. And so to continue along this line a little bit, also based on your long-term questions, right? So what kind of exercise regimen, for what kind of applications? You mentioned different type of chronic conditions that may benefit from different type of exercise. So since you are the expert here tonight, right? So what would you envision would be, for instance, a long-term healthy longevity exercise routine, if you want? So would it be an alternation of high intensity exercise with endurance exercise, and how much would have to be tailored to different individuals, for instance?
I love your question, Vincenzo. And I think just from a purist standpoint, right? We see in the popular press where people are gravitating towards one thing to the next. First day it was HIIT, the next day it is strength training for sarcopenia. Another day in the 1970s, it was aerobic exercise. So now that we understand a lot more on the systemic physiological adaptations, we understand a lot more on the molecular adaptations, I think it's safe to say that actually it's a combination of different types of exercises that would then drive adaptations at the cardiac level, at the musculoskeletal level, at the immune level. Because, as you see in our very preliminary study, we show that if you were to just do a certain kind of exercise, you elicit different responses, just speaking in terms of exerkines alone. So then we know that resistance exercise, for example, has the biggest benefit on bone loading, on preserving muscle mass. And now others have also shown that they may also play a role in glucose disposal or improving some aspects of cardiovascular health. But it still doesn't take away from the fact that, certain exercises may not be practical for the general population, or simply not sustainable in the long term. So we run into the question of practicality. Not everybody wants to go to the gym. Not everybody wants to run. So then I thought about this, and I think then if we were to distill exercise or physical activity down, then it just becomes all about human movement. And then we consider other kinds of human movement, like dance, ballet, right? Or even other things like martial arts or capoeira, right? All of these different human movements are essential to health. But people have different interests, people have different passions, and what works for one may not work for others. I think the literature is rich with aerobic exercise or strength exercise simply because it's easier to control. And for other exercises, it may not be so easy to actually define a protocol to measure that operationally. So I think I guess it's a very lengthy answer to your question, but I think coming back to it, it just means that we need to understand how movement in different forms and variations affect physiology, both in a systemic sense and in a molecular sense.
Right. Yeah, I think these are, as I say, studies that would be very fascinating to run if you can run them all, right? So studying which thing to prioritize the first would be quite important in this case. But maybe still then coming back to the fact that you mentioned also, again, the importance of movement in general, but also, different people age differently, and some of them, for instance, suffer from, as you mentioned, sarcopenia, which in Singapore, by the way, is quite prevalent. And that's also to some extent, one of my research interests. But in your case, coming from the exercise perspective, and given also your example of the populations that you mentioned in the beginning, that they are basically moving around all the time, right? And they are eating different things and so on. My next question is, where do you see nutrition, for instance, also integrating with the exercise? Is it something that will certainly have a combinatorial or synergistic effect on exercise? And do you have particular tips or recommendations or ideas on how you would do this?
I think it's a tricky one. I think the fact that you mentioned diet, and we say in Singapore that people love to eat, right? Or we live to eat.
Right.
So then the thing is, how do we combine an effective use of nutrition in the face of so much temptations? I think in terms of dietary control, I think if we were to go back to what are the guidelines that are given, um, there is, I think, a overconsumption of some of these high-fat diets or a lack of fiber intake in, I think, the majority of the population. So when we look at that, it's not surprising because when you go out for meals, most of the options for vegetables, the portions are measly, right? So there's an overabundance of carbohydrates and an overabundance of animal protein. And then if we were to take into account the different methods of preparation, deep frying, for instance, or the addition of various condiments, all of these would add, and going back to what I was mentioning earlier, though all of these would add maybe not maybe, it would add an inflammatory load to the system, just to put it very bluntly, right? And if we were to do even all the exercises that we wanted to, it's not going to erase some of this detriment. So then, if we were to have the healthiest diets, it would literally be what, again, our ancestors used to have. Very minimal portions, small portions, mainly vegetables, very little meat,
So
very little processed foods.
exactly. So control of calories and trying to eat healthy
Absolutely.
as a general recommendation for now, and then trying to investigate what particular nutrients are required for particular population targets, I guess.
Absolutely.
And regarding more specifically your study, right? So one thing I noticed, for instance, especially in the last study where you're doing the measuring of the irisin after exercise, indeed, you mentioned that different type of exercises were impacting differently, maybe, or enhancing or decreasing this difference between elderly population and a younger one. But I have the impression that although the cohort is small, with the high intensity exercise, even the baseline was not really different, right? And then it sort of stayed stable for the aged cohort, right? So do you think there is a particular explanation for that, although the cohort and the numbers are very small, right? So I was wondering if you have an explanation or speculation for how irisin would respond to the different type of exercise and especially in the old cohorts particularly.
Certainly, Vincenzo. And I think, again, maybe this boils down to, again, the myokine response. And if we think about the myokine response, I think it gets very interesting because we assume that muscle is homogeneous, right? But then there are different proportions of the type 1 slow twitch fibers compared with the type 2 fibers. And as we age, we lose a lot of our type 2 muscle fiber types. And then the question is, and even this is not yet known, is, what are the types of muscle fibers that are most responsible for releasing specific types of myokines, right? At least from the literature, we know that type 2 muscle fibers may not be as responsible as type 1 fibers. So then I think this discrepancy between muscle fiber types would come into play, where at rest, the concentrations of irisin in this instance may not be different, but it's during muscle contraction where you're really recruiting the different muscle fiber types that could elicit the response of irisin release that could actually be different because simply that in older adults, for example, there are less type 2 muscle fibers relative to type 1.
Would that also have a possible explanation why in the aged, you said there was more correlation with muscle oxygenation and irisin levels only in the elderly cohort, right? Is that particularly because of the type of fibers also that are more accessible to some extent or differently accessible? Or for instance, and what do you think of the vasculature changes during exercise in aging? Can we also target those, for instance, maybe together with trying to maintain or raise the levels of irisin?
I think that's a great point, Vincenzo. And I think that in terms of the high muscle oxygenation, perhaps it's really driven by the energy demand. I think maybe because of the decrease in muscle mass and maybe in type 2 fibers, that the older adults are simply consuming more oxygen per unit of muscle fiber. And I think that's why there's such a strong correlation. But whether or not, we can say that this is a direct result of the muscle fiber type 2, the release in the irisin response, I think, again, those are correlational studies. And unless we do animal studies to probe this, I don't think we get a very clear answer yet.
And so regarding, again, the question on the vascular, I think I have time for one or two more questions.
Okay.
Is there interventions, maybe this, like of my experience from the clinical perspective, have people tried already, there are different type of drugs that are considered, not allowed during sports and competition, right? So doping. Are some of these actually able to affect the vasculature and therefore the muscle performance? And would that be actually something, I don't know, interesting to consider for elderly that want to maintain, let's say, a sort of healthier muscle health, but combining it with ways to promote oxygenation of the muscle, for instance?
Sure. I guess I could maybe think of, like beetroot, for example, because it does increase nitric oxide levels. And, there are instances where athletes actually consume beetroot juice to actually boost angiogenesis or, growth of blood vessels, so at least inducing nitric oxide production, right? But again, the question then is at some point, is there going to be a ceiling effect? And if so, would it be more effective in older adults than in athletes? I guess I'm not familiar with the literature enough to comment on that. But certainly, I think it would be worthwhile to try some of these types of ergogenic aids.
Right. And perhaps one last question before we pass the word to Atika. What about, in the exercise intervention? I think it's already pretty established that things like, we're looking at biomarkers or different ways to monitor changes with exercise. Is myostatin something that is, for instance, downregulated with exercise so that then you could downregulate that signaling pathway in order to maintain muscle health for longer? Is that something that emerged from also the acute exercise from Schneider lab, for instance?
That myostatin, that story is interesting, but I'm afraid I haven't actually followed that for a while.
All right, so it could be something to
Could be possible
to look also maybe in your own clinical studies here, right?
That would be a good point. Yeah.
All right. So I think we have now time to pass to the Q&A. So I will pass the word to Atika, who will moderate the session.
Thank you very much, Dr. Jorming and Dr. Vincenzo, for the insightful sharing session. So we have many questions from our audience today, and let's dive straight into them. So the first question would be, based on the slides showing various types of exerkines, like myokines, adipokines, and hepatokines, could you discuss whether different forms or intensities of exercise preferentially stimulate the release of specific exerkines, and how this might influence targeted strategies for preventing age-related disease?
I think this is a great question. And again, it boils down to what are we measuring and what kinds of exercises are we actually implementing? So the short answer is we still don't know enough, right? We know that some of these releases of these exerkines may be specific, let's say for improving immune function or to dampen inflammation, but those are only done in very specific controlled conditions. And again, not a lot is being done in other chronic diseases to know whether it was specifically these types of exerkines that are responsible for specific protective effects. So the answer is we still don't know enough.
Okay, thank you, Dr. Jorming. And the next question would be, why are there fewer women represented in scientific research?
Okay, another good question as well. And we tried to address that in our first study where we recruited women. Essentially, it's because of the regular menstrual cycle. So because of that, there's a lot of fluctuation in hormones that need to be controlled. And it needs to be, if we were to run any kind of exercise trial intervention, then we need to account for the same time of testing or the same week of testing. And just from a scheduling standpoint, it's more logistically difficult to get women to come in, let's say, at a specific time during their menstrual cycle. In our study, what we did was we actually recruited women during the first 3 days of menses. So then everything is standardized to that first 3 days of their menstrual cycle. Then at least things are more standardized in that sense.
Okay, so another attendee here asks, what do you think of EMS training? Is it scientifically proven to be effective?
So EMS is essentially electrical stimulation of muscles, right? It's essentially used to allow muscles to contract. And I think the thing is that it is useful in terms of rehabilitation, from a rehabilitation standpoint. Let's say if a person is unable to bear load, carry his or her own weight, then EMS potentially may have some benefits in terms of maintaining some functionality of the muscle, of the injured tissue. But from a long-term standpoint, EMS, at least the evidence shows that it hasn't been used to improve, human performance or exercise performance. Okay, I think it's a good rehabilitation tool, but not necessarily useful in terms of trying to improve health.
Okay, and then we have another question here. This attendee is asking if do both aerobic and resistant exercise reduce inflammatory indicators, and which type of exercise is more beneficial for the young versus middle age?
So there are two questions asked by this person. So the question is, number one, whether aerobic or resistance exercise is more beneficial in reducing inflammation, right? And the second question is whether it's more effective in the young or the old. So if I were to address the first question, aerobic versus resistance exercise, we still don't know enough, all right? We still haven't designed effective interventions to be able to understand whether they're effective, if similarly effective in terms of reducing inflammation. And the nature is that exercise itself is broad, and even aerobic exercise itself is broad. There's low, high, moderate intensity. There is weight bearing versus non-weight bearing. So for example, cycle exercise versus running versus versus swimming, for example. Now, if we talk about resistance exercise or strength training, all right, there's also going to be different aspects of strength training. Is the person training for strength, so purely just getting very strong? Is the person training for the muscles to get big? Okay, or versus the last point is, are they training for the muscles to withstand multiple lifts? So that's more muscular endurance. So within each broad spectrum of exercise types, we are already seeing different permutations, right? And when we do scientific research, we usually want to compare things that are very clear-cut, things that are very distinct. So for example, comparing low aerobic exercise intensity to high aerobic exercise intensity, so that we know exactly what modality we're comparing. If we compare strength exercise to aerobic exercise, then the difficulty comes in to say, are we going to make sure that they're all exercising for the same duration or the same intensity, and how do we quantify that? So there is no easy answer to that, okay? But long answer short, any kind of exercise would have an anti-inflammatory effect. And I think, at least just from skimming through the literature, we would say that most of the studies have been done in aerobic type of exercise, so running, cycling, okay? And then now I think more research is gaining in the area of resistance exercise, so strength exercise. Now, in the young and the middle age, if we are talking about reducing inflammation, then it's evident that as we get older, then some of these pathways do get broken down, and inflammation will be higher in middle-aged people compared with younger people. So from that standpoint, then aerobic or resistance exercise would have a better effect or greater effect on inflammation. I hope that would answer the question.
Okay, and then we have Steve from the audience asking, what are the perceived functions of the different novel exerkines that you're measuring?
Yeah, so a shout out to Steve. Hi, I know Steve. Some of those functions of the exerkines would involve things like triggering the release of BDNF, which is brain-derived neurotrophic factor, which could cross the blood-brain barrier and induce neurogenesis. So it would have improvements on our cognition, for example.
Okay, I think we still have time for a few more questions.
Okay.
Okay, the next one would be, given that exerkines are released by multiple organs and have systemic effects on cardiovascular, metabolic, immune, and neurological health, are there any specific biomarkers or exerkines that clinicians can actually monitor to assess the effectiveness of an exercise program in promoting healthy longevity?
I think the answer is okay, again, a short answer to this is we still don't know enough, and we need to correlate the effects of exercise on exerkines with some of the more functional outcomes. So meaning that, let's say, if we want to look at things like improvement in muscle strength, then we need to look and see what are also some of the biggest changes in the exerkine response. Is the concentration increasing? Is it decreasing? Are they significantly different before and after the exercise intervention? So these are some of the more correlational kinds of analyses that we can do, but they do not necessarily drive the explanation of whether the increase in exerkines could directly impact the functionality that is also improved.
And also, would it be sufficient to be physically active day to day, like doing housework, going on public transport, or even walking to work without intentionally exercising?
I think, again, this question from Claire is meant to say, would it be sufficient to actually maintain health? Yeah. So if we were to take it as being physically active, then that is, in my opinion, that would have to be the basic, that would have to be the baseline. So in other words, if people were sufficiently active, then there may not be a need to exercise intensively. But when we talk about the kinds of physical activities today, when I look at these comments, right, the level of muscle contraction may be too low to induce any kind of medicinal effect. All right. So, excuse me. So for that matter, it is important that some level of higher intensity activity is actually implemented so that the organs are actually able to then withstand a higher amount of adaptation. If we were to just go back to my illustration of the the hunter-gatherers, right? The hunter-gatherers are already 2 to 3 times more active than most of our modern day societies. And in that standpoint as well, when the hunter-gatherers do engage in activity, it's usually bursts of high intensity activities such as them hunting or foraging, which is several folds higher in terms of energy demand than the kinds of things that we are doing in our day-to-day lives. All right, so it is better than being sedentary, but in terms of really truly activating that medicinal value, I think it's still below par.
That's interesting. And the last question would be, are there any known chemical compounds that can stimulate irisin production?
I must say I'm not familiar with that. So I'm not too familiar with that, unfortunately.
Well, then I think we can close with this, and that means we maybe while we remember or find out which drug is doing that, we still have to be hunter-gatherers, I guess, to make sure we have a high irisin enough. So thanks again, Jorming, for joining us. It has been a pleasure, very informative talk. And I want to remind all the participants to use the chat function and choose the panelists and all attendees option to leave the comments and the feedback so that everyone can view it. And also regarding the next events that are going to happen, we have the Healthy Longevity Intensive Course 2025, which is going to be in June 30 until July 11th in the University Town in NUS. And also the Healthy Longevity Talent Incubator, for which the inscriptions for the applications are still open. There is basically, this event consists of 10 days of intense interaction. You can learn a lot from other scientists that are going to be there presenting and exchanging with them. And therefore, it makes up for a very, let's say, fruitful and unforgettable experience. I've been, let's say, myself, one of the lecturers there, and I can say it's a very, I would say it's a very recommendable experience. So don't miss your chance to be part of this and to learn more about neuroscience and healthy longevity. And then I want to remind you about the next Healthy Longevity webinar, which will be on May 8th and not on 1st, because May 1st is public holiday, and the topic to be still confirmed. And for today's session, there is a short reminder that it's never too late to start exercising. This will be part of the first video, so if you begin today, maybe this is you that will be at 85.
I am Ernestine Shepherd. I am 85 years of age, and guess what? I've still got it.
Still got it? Indeed. This octogenarian athlete refuses to let anything stop her.
My mantra is determined, dedicated, disciplined to be fit. To get out and exercise, I don't let anything get in the way.
Ernestine's journey to health and fitness started when she was 56 years old, with a promise to her sister Velvet shortly before she passed away.
She held her head up and said, I want you to promise me that you will continue to help others to live a positive, confident lifestyle.
So Ernestine started working out daily, and at the age of 71, she took her training to the next level and entered her first bodybuilding competition.
I was the oldest one there. Next thing I knew, I came in first place.
Then came a call from the folks at Guinness World Records.
I was the oldest female competitive bodybuilder in the world. It wasn't just wonderful for me, it was wonderful that I was fulfilling her dreams and then plus helping others.
Even now, Ernestine does not show signs of slowing down.
Monday, Wednesday, and Friday, I'm at the gym at 7 o'clock in the morning. I have done 11 marathons. And just 3 weeks ago, I did a half marathon. I don't feel like I'm 85. Age is absolutely nothing but a number.
I'm feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down.